All right, well, why don't we go ahead and get started? So just a reminder for everybody that this is being live streamed and recorded, and that the grants supporting this were from the NIH and NSF for the computational platform in general, and then EPA and ORISE for the specific projects I'll tell you about. So my name's Shane Hutson. I'm at Vanderbilt. I'll tell you a little bit about a project I worked on when I spent a year at the EPA's National Center for Computational Toxicology, working with Tom Knudsen and his group there on their virtual embryo project. And this was one where we were trying to develop a CompuCell 3D model of secondary palate fusion and how that goes wrong, and a few mutant examples, and in at least one sort of toxicant exposure that leads to a failure of palate fusion. Let me find the right place to click. Yeah, so this paper is out. It's in Chemical Research and Toxicology, so all the details are in there, but I'll give you the highlights of it as we go through here. And if you want to go back and get the fuller picture, that's always available to you. All right, so take-home messages, what we're trying to do. We're trying to model fusion of the palate, right? The soft palate, which is the top of the mouth. We're doing that with a number of different cell types, and we'll go through all of them. A biochemical regulation circuit that's sketched out here at the top left, and we'll go through that in detail too. I'll highlight the important parts of it. But you can see screenshots here, a little time-lapse movie of the cells as they grow, meet in the middle, and then fuse into a single tissue. And we'll talk about all the details of how that happens. One of the important things we had in terms of matching this to experiments, right? How do we know that there's some ground truth to it? One is sort of just the timing of all the events and getting them right, but then recapitulating genetic knockouts, that we can go in and modify a line in the code that would mimic knocking out, for example, Sonic Hedgehog there, SHH, or BMP4, and that we would get an in silico phenotype that matches up what you see in the actual organism. And then finally, since I was working with EPA on this, we want to use it for developing some mechanistic insight into developmental toxicity. And in this case, we were looking at dioxin. That's a classic toxicant that is known to disrupt palate fusion, one of the many things that it can unfortunately do. All right, so we chose palate as our fusion model, but just to remind you that fusion is everywhere during embryonic development, right? Your diaphragm grows in from both sides of the body and then fuses in the middle. There's fusion going on in heart development. Your neural tube curls over and then fuses together into a single tissue. Optic cup, urethra, all right, there are examples all over the place. So it's a very classic thing that has to happen where you have two tissues that have to merge into one. When we're looking at the palate, right, normally there's two palatal shelves that grow down beside the tongue. They're labeled with P there and tongue with T there in the middle at E13.5. These are days, embryonic days in development of the mouse. That was the model that was used here. Those shelves of the palate flip up above the tongue by about day 14. They grow towards one another and touch, make contact, and then that midline between them is going to have to disappear so they become one uniform tissue. Right now, if we take a little closer look at what some histology of this process looks like, here in A and E, you can see two palate shelves growing towards one another. And you can see just a little bit of difference in the staining here. It's easiest to see in B and F in the middle. The sort of blue area are mesenchymal cells, and then the browner area are the epithelial cells on the outside of that. And one of the keys to fusion is that after the two tissues meet, the epithelial cells that are in the seam need to go away, right? And they could go away in a number of ways. They might migrate, they might die, they might change into another cell type. Right, all of those are possibilities. Some other views here. This is in C, looking up on the palate from the bottom. So you can see that MES is the midline epithelial seam. And in D there, it's a little side view of it again. And you can see that as the seam resolves, you end up oftentimes with these little pearls of cells left in the middle, and those go away over the next day or so. during development. So that's sort of what we want to try to match up and reproduce with the model. So another view of it, right? This is really a 3D process. We're going to treat it just in cross section. If you look at that middle panel there, D at 14.5 days, you can imagine taking a cross section that goes right through both of those PSs, and that's about where we're modeling it, right? And you'll see that. And so the cross sections will look much more like what we have down here at the bottom. And we're going to model the outgrowth of the shelves, their contact, and then their fusion. We're not going to worry about the shelves flipping out from above the tongue. We're going to start past that point. All right, so in terms of a sketch where we're going, I'll give you some idea what's in the computational model, what our thinking was behind how it was organized, what the biophysics were, what the biochemical regulation is. We'll look at the simulations of wild type and some in silico knockouts. That'll be one of our ground truths that we have to match up. We'll look at simulating a toxicant effect, dioxin. And given some time, we'll take a look at the model code, and there's some code writing exercises that those who are in here can do, those who are watching on YouTube can access later. All right, so sort of the players. There's a lot of different cell types that are involved here. And so I'll be careful in trying to describe them so everybody knows what we're looking at. So we've got several different types of mesenchymal cells, right? The sort of cyan that you see in the light green, these are both mesenchymal cells in the palate. We characterize them as two different cell types because there's sort of a nasal side and an oral side to the palate, and they have different behaviors. So the cyan ones are the nasal side, and the greenish ones are the oral side, mesenchymal cells. This sort of peach color that's running around all through and around these mesenchymal cells, that's the extracellular matrix of the mesenchyme, right? And so these cells are embedded in that, and we treat it as a cell type, even though it's not really a cell, right? It's really matrix, but we treat it as a cell, and I'll show you how it behaves in relation to the mesenchymal cells that are in there. The bluish cells are our epidermal cells of various kinds, or epithelial cells of various kinds, not an epidermis, epithelial cells of various kinds. And we compartmentalize those, right? So there's basal epithelial cells, and then there's another part of matrix, the basal basement membrane, that's here in sort of this maroon color, that's going to be treated as a cell as well. It'll come into play there. And then over the very outside of this, there are periderm cells, and those have apical and basal compartments that go together and form one polarized cell on the very outside of this. Right? And by all means, stop and ask questions. Yeah, James put that in the chat. By all means, ask questions. Stop me as we go through if you have any. All right. At the basic, the heart of a CC3D model is Metropolis Monte Carlo Potts model. And so you have to define everything that goes into the Hamiltonian, right? The energy functional that defines how things behave here. The ones that we're using in this model are sort of your basic ones. There's contact energies between all the different kinds of cells, target areas and target perimeters for each cell, since we're doing this in 2 dimensions. And a few, in particular, the periderm cells out there have a target length because we want those to remain very squamous. And between those, you can define the properties of the cells that will give you the more or less correct biophysics. So now, in terms of the contact energies, I had a student this summer that I was training. We're trying to do a wound healing model, and he was like, well, what do I pick for the contact energies? And this is the matrix that we use for this one. I can't swear that these are the absolute best contact energies. I can sort of tell you the hierarchy, right? Which ones should be higher than which other ones, but I can't tell you exactly how much higher they should be. And a little bit of that is empirical, playing around and figuring out which ones work. But just to show you the kind of decisions that go into it, if we just look at that yellow highlighted block there, these are our mesenchymal cells and the matrix that they're surrounded by. You'll notice that the mesenchymal cell matrix contact energy is actually negative. So the simulation as it runs is going to want to increase the length of those sorts of interfaces. And that's why we get these very ragged boundaries of the cells, and why there are not cell-cell contacts, but cell in contacts with matrix, right? Cells are sort of surrounded by matrix in that grouping. The cell-cell is, a little bit positive, but not too high. So it's not all that unfavorable for these kinds of mesenchymal cells to be in contact with one another, but it's not as favorable as them being in contact with matrix. This is one way to get this sort of, I'm blanking on the right word for it, but cells that look like they're reaching out extensions in various directions, right? Now, in contrast to those cells, if we look at the cells that are on the outside of this palatal shelf and what they're doing, we have epidermis and then the two types of periderm compartments. If we look at these, and in particular how they interact with the medium, right? You'll see there that it's really unfavorable for epithelial cells to be in contact with the medium, fairly unfavorable for the basal part of periderm cells, and then not quite so unfavorable for the apical part, right? So the apical part of the periderm cells is going to end up on the outside. And you can sort of go through, all right, if those are on the outside, what are they going to be in contact with? Well, their best contact energy is with periderm basal compartments of the same cell, unfavorable for periderm compartments of the neighboring cell instead. Pretty favorable to be in contact with other apical periderm cells, so they'll form sort of a boundary layer there. And then once we get the periderm B cells, those are then reasonably favorable to be in contact with epiderm. So it's sort of that ordering determines how they're going to structure on the outside of the patch there, and how they're going to arrange themselves. Now, the biochemical circuit that regulates this, everything that's in a white box there is a chemical compound that we actually have cells secreting and run a diffusion field for it, and we track exactly how much is where in space and time. Some of these are just accessories. Some of them are really at the heart of what's going on. And so there's two really main loops that are in this circuit. One is this feedback between sonic hedgehog, SHH, and fibroblast growth factor 10, FGF10, and they are mutually reinforcing. So the epithelial cells secrete sonic hedgehog, and that then drives the mesenchymal cells to secrete FGF10, which in turn drives the epithelial cells to secrete sonic hedgehog. So we have this positive feedback loop between those, and we get really strong areas next to one another. They're secreting those two compounds. And all of these, right, we know a lot about the pathways by which these act, right? Sonic hedgehog interacts through patch and smooth and through GLI1. Those aren't explicitly in the model. They're here because we know that pathway, but we're really just linking then the fields that are on the outside of the cells to cell behaviors, right? And so, for example, sonic hedgehog impacts BMP2 secretion, and that directs proliferation and matrix secretion. And it also, FGF10 is linked to proliferation. So we're going to have this positive feedback loop where the mesenchymal cells in the palate and the epidermal cells in the palate are both encouraging one another to grow. And so they'll grow together and the palate shells will grow, right? And keep the right amount of epithelium for the right amount of mesenchymal cells as they go. The other key part of this is this negative, this mutual inhibition part over here, where epidermal growth factor EGF and transforming growth factor beta-3, TGF beta-3, mutually inhibit one another, right? And that's going to act like a switch. One or the other will go high and the other one will go low. And if we started them out even, it would be completely random which way it went each time. But these then create, excuse me, control behaviors of the medial epithelium, right? So if EGF is the one that goes high, then it encourages proliferation of those cells and suppresses their apoptosis, their transition to epithelial mesenchymal transition, their fate change, and it decreases their motility. On the other hand, if TGF beta is the one that goes high, it encourages those cells to die, to change into mesenchymal cells, and to move, and it suppresses their proliferation. So that switch is going to be really important for the behavior of the system. And I labeled in here epherin B with a question mark, because really what we don't have epherin explicitly as a molecule, but we have some contact dependent behaviors, which is mimicking what epherin would do in the system, right? It's a juxtacrine signal instead of a paracrine, so it's really contact driven. Questions so far? Okay. So I'll let this play. There's some still frames over here on the left, and hopefully the video will transmit reasonably well that you can see it grow. We have two palatal shelves that start out here left and right. If you want to ask questions later, we actually do start the simulation at this point. Well, this is 100 Monte Carlo steps in, but it hasn't changed too much, by defining what's called a PIF file, a POTS initialization file, where we define exactly which lattice sites belong to which cell, right? And we set that up to begin with. And then as those grow and make contact and then fuse with one another, you could see, and I'll let that play again, watch how the cells in the middle, the midline, disappear. Some of them die. Some of them migrate into those triangles and go to the top and bottom epithelia. And a few of them, these red cells in the middle, they underwent an epithelial mesenchymal transition, and we tracked them by having another cell type that we just colored a different color so that we could see which ones actually came from the epithelium when they started out. And you'll notice that as it closes up, there are some of those little pearls of cells that are left for a few minutes before those disappear as well. All right, so far, I mean, this took a few months to develop and think about how we could reproduce the normal behavior. We can look at all the cell fields that are in there, right? I said that we actually track 9 of these different secreted factors. And you can see how each of these behaves as a function of time. I'll play it, and you can pick your favorite one to take a look at while it plays. Whoop. Actually play it there. And you can see that those are dynamic, and they change with time as the simulation runs forward. And we'll take a closer look at how they are patterned in space and time. But what you'll notice at first is that, right, there's a different spatiotemporal pattern for almost each one of these, right? They really are controlling the cell behaviors in very different ways and in different patterns. So if we take a closer look at this, and we compare what's going on in the model, right, to what's seen in experiments. So sonic hedgehog is expressed by the epithelial cells in the medial part and in the oral part. And here's a label of what's actually seen in a palate. It's a little bit concentrated on the oral at this section, but there's a little bit towards the medial as well. This other labeling shows more on the medial, so that's approximately where we want it. If we look at FGF10 or BMP2, those are both spread throughout the oral part of the mesenchyme. BMP4 is pretty much everywhere in the mesenchyme, and that's where we have it in the model. FGF7 is in the nasal part of the mesenchyme. Noggin is expressed in the oral part of the epithelium. And then TGF-beta is expressed in the medial part of the epithelium. And it diffuses out, right? Because it ends up impacting what happens in cells in the mesenchyme as well. So we've got approximately the right spatial distribution for all the chemicals of interest that we were tracking. And we've got approximately the right cell behaviors as a function of time. And so you can see how these things would just focus on that positive feedback loop between sonic hedgehog and FGF10. You can see that they sort of localize together or next door to one another, and eventually end up just in the oral part of the mesenchyme. Or we can track TGF-beta. Well, let's see if we can go back to the start on that one. Maybe it'll let us do that. Nope. All right. Movie's not there for TGF-beta. We'll see TGF-beta in some other places. But then one of the important tests was to do some in silico knockouts. So in this case, it's easy to do the ones for the secreted factors because we just eliminate that secreted factor. and see if we've got the programs down, the behaviors downstream of that more or less correct. So if we knock out Sonic Hedgehog, we've knocked out that positive feedback loop, and the shelves basically just don't grow. And so they don't make contact. If we knock out FGF10, it's a little bit less of an impact, but the shelves don't grow sufficiently. They make contact, but it's not really complete. BMP2 doesn't grow enough for the mesenchyme. BMP4 has this similar sort of not quite enough growth as FGF10. And then there's others like FGF7 and Noggin that don't really seem to have much of an effect. They're sort of accessories that fine-tune the system, but they're not absolutely required to get the cell behaviors. Other ones like TGF-beta and EGF, right? This is the mutual inhibition that takes place in that epithelial seam. And if we don't let TGF-beta be present, right, we knock it down, then we get this huge overgrowth of the epithelium, and it never goes away, and the tissues don't fuse. If we knock out EGF, the system's okay. Actually, this turns out this is exactly what happens in the organism. EGF mutants are pretty much okay. TGF-beta 3 mutants are definitely not. Their palates don't fuse. We can even knock out matrix metalloproteinase, and you can see that there's a whole bunch of basement membrane left in there that just doesn't get degraded when it's supposed to fuse together. Yeah. And this is the TGF-beta mutant movie playing, and you can see how just the epithelia overrun. They don't suppress their proliferation enough because the TGF-beta isn't there. And then what we wanted to do was to link the model to toxicology. And so the classic one for palate toxicology is dioxin, TCDD. Right, this is generated in burn pits more or less nowadays because no one makes it and uses it on purpose. But you burn organics and you get some of this. Its mode of action is that it binds to the aryl hydrocarbon receptor. And in the palate, what it does is it suppresses TGF-beta signaling, right? And you can guess from what we saw on the previous slide that if you suppress TGF-beta signaling, it'll be similar to knocking that out. And so we get changes in apoptosis, proliferation, and differentiation of those epithelial cells. The medial edge doesn't degenerate like it should, so we get altered fusion and cleft palate. And so then in terms of putting in that toxicant effect, we had to decide where we were going to put it in our biochemical model. And so what we did was just have it, based on the biochemical evidence, what's out there on the literature, is have the aryl hydrocarbon receptor affect this part of the loop. And what we specifically did is have it affect the amount of EGF receptor that was present. And by changing the amount of EGF receptor that's present, it changes whether that switch is likely to go to EGF high or go to TGF-beta 3 high, right? I'll show you some of the diagrams where you can plot out exactly at what suppression or increase of EGF receptor will cause that to happen. Yeah, so here it is, right? So what we have in terms of the biochemical model for how this is going to play out is that the making of EGF, right? That's what the little right turn arrow represents there, right? When EGF is made, it might be degraded at some rate. It binds to the EGF receptor. And if it binds to the EGF receptor, that complex inhibits the production of TGF-beta, right? TGF-beta is degraded at some rate. If it binds to its receptor, that receptor complex inhibits the production of EGF, right? So that's how it's encoded in the model. We do this with a few Hill functions, basically. You can easily program these inhibitory interactions. And depending on the parameters you pick for them, you might end up with something like B, which is a low hysteresis switch. So there's conditions where the EGF receptor is low, and so it favors TGF-beta. You get a little bit above 1 in terms of the fold change of the EGF receptor, and this thing all of a sudden switches very sharply and drops down here, and EGF is favored over TGF-beta. Okay. You can also, depending on how you choose the parameters, have a very high hysteresis switch. And so if you increase the EGF receptor, it's pretty stable to have TGF-beta 3 continue high for a while, and then eventually drop down and EGF go high. But if you then decrease the EGF receptor, it wouldn't immediately switch back. It would switch someplace lower, right? And so you get this hysteresis in the system that depends on which way you're trying to go with the switch. It's got some history dependence to it. And so, for example, this is like a constant exposure to TCDD or dioxin with the low hysteresis switch. And it only required going up to like 1.15 times fold increase of the EGF receptor. And if we play this, right, we get a little bit of remnants in the seam, but it more or less fuses it sort of right at the edge of it. If we make the high hysteresis switch with a little bit higher fold change of EGF receptor, we get a little bit more of the epithelial seam that sticks around for a longer time. And even if we get transient, right? Suppose there was a dose of dioxin at one point during development of this organism, but then it went away, right? It was cleared. And so we get a fold increase of EGF receptor, and then we bring it back down. Well, if we're in the low hysteresis switch regime, well, then that's fine. The palates don't fuse at first, but when dioxin goes away, then we can go back and fuse the palates together. So it's delayed, but not prevented. On the other hand, if we have the high hysteresis switch, it can get stuck in that unfused state, right? The seam never went away, and we can't switch back because it's just so much hysteresis and history dependence that's in the system. So one of the things that was really critical as we were doing this is that we had to have the epithelia be polarized, right? To have compartments in the cell. So there was apical and basal parts of it. In this case, we had apical and basal periderm, and we had another layer of epiderm or epithelial cell below that. And that was important because at first, when we ran it and just had a simple layer of epithelial cells on the outside, the palate shelves would meet in the middle and they would fuse every time. We couldn't prevent them from fusing, right? And so that told us there was something that wasn't quite right about the biophysics in the system, and it really turned out to be the polarization of the epithelial cells. They were really critical to making sure that they wouldn't fuse. And so we can take a look at the code, and I can show you how that's done, and we can look at some of the exercises to take a look at how you can make a polarized epithelium. Okay. So let me see if I can, let's see, I will probably have to stop sharing for a second and pull up something different to show the code. Let's see, where is, let me just show the desktop, and then I can show whichever one we're looking at. We're here at 20. All right. So can folks see the code window? That's what's showing appropriately on the top. All right. So I'm going to go to the palette model steppables, and there's a whole bunch of them here that we ran. But let me go down a little bit. Sorry for the fast scrolling. So we have this class, palette model cell behaviors, and this is what we're using every step through as we go through it. And it's going to control all the behaviors that we have downstream of those chemical fields, including the secretion of the chemical fields. And one of the things that we have, let's see. Let me just do a search and find the one I'm looking for. Here we go. So we have a steppable that's just manage epithelial polarization. And the reason I wanted to pull this up, I won't show you the whole thing, but I'll just give you an idea where to look for it in here. So you can imagine one way to do this simply is at the very start to define a layer of basal paraderm cells and a layer of apical paraderm cells and have them be in the right orientation with respect to one another. One of the difficulty with doing just that is it'll work okay, but if, for example, a basal compartment or an apical compartment dies, which happens during the simulations, then you end up with a cell that no longer has matching apical and basal parts to it. And so in terms of you have to actively maintain the epithelial polarization. So what this steppable does is it looks like it's set for every one Monte Carlo step. It goes through and checks every one of the basal paraderm cells and says, does it have a matching apical part? If it does, great. If it doesn't, then it nucleates a new apical paraderm part for that particular cell. Same thing with checking the apicals to make sure each one of those has a basal part to it. Right? And that way the system constantly maintains having both apical and basal compartments, right? Those cells maintain their polarization the entire time. Some of the examples, when we go through and try those, you'll see that there's new epithelial cells that are made, and those epithelial cells, when they're made, recognize that they don't have an apical compartment, and they make a new one immediately right after that. And so it's important for the maintenance of it to do that. Any questions?
It's really not a question so much, but just a clarification. Shane, at one point you wanted to track a particular cell transition, phenotypic transition.
Yep.
So you made a special cell type just so you could see that cell.
Yeah, that is what we did. Yeah.
Is that cell type that you made identical to an existing cell type?
It was a cell type that was made just to track. Right, it was exactly the same as the oral mesenchyme cells. Yeah.
So that means you had to go back through the adhesion table and
another line in the adhesion table. Yeah, just repeat it.
And it'd be easy to make a mistake and change one of those lines, then not the other, et cetera. So wouldn't it be really handy if CompuCell had a way so you could say, I have a cell type that's identical to another cell type, and it should just inherit everything from that other cell type, so that I don't have to add into the adhesion table.
Yes, inheritance like that would be wonderful. Yeah.
Because the other problem I see often, and I think you've got it in this simulation as well, is that you have multiple cell types that are very similar. And really, they could have the same adhesions or very close to the same adhesions.
And they do. For example, our two types of mesenchyme cells, the oral and the nasal parts, right? They have the same contact energies. They're only distinguished by their responsiveness to the chemical fields.
Yeah. So, it's real tedious and error prone to make a new type that's almost identical to an existing type. Because then you always have to remember, well, if I'm going to tweak one of them, I got to remember to tweak the other one, or now they're no longer behaving the same way.
That's right.
And it looks like to me, so in this, your Steppable, there's 2,000 lines, 2,800.
Yeah, at least it probably goes. Let's see, 2,800.
Yeah, it's down in the scroll bar in the bottom. Yeah, okay.
Oh yeah, yeah. So there's a lot in there that required significant code. So.
Are you done?
I am.
Okay, I want to bounce something else off of you. So you've got this beautifully complex model, which is complex for a model that probably still 5% of what biology is actually doing, which is fine. But that's life in the modeling world. You have certain defects you want to generate, and you think you have an idea of if I tweak this, it's gonna fuse, but not eliminate the epithelium, the extra epithelium. You have various other things that observable failures and possible mechanisms that lead to that failure. And so you tweak the model to try to I think it's this factor that's going to do this, and it does or it doesn't. And if it doesn't, you come up with some other ideas. If it doesn't, you might just say, well, the model's not right. The fundamental model's not right, as opposed to the model's right, but my mechanism's wrong. So anyways, you open up a huge can of worms of just because this change in one parameter resulted in this flaw, it doesn't mean that's the only way to get that flaw.
That's right. Yeah, absolutely.
So James and I, when we rarely have time to talk, have been talking about trying to take that problem and turning it around the other way. Instead of saying, here's the normal system, and here's the known flaws, how do I build a model that reproduces both? To here's a normal model. If I just randomly change stuff, can I classify and group all of the outputs the model produces?
Oh, okay. Yeah, that's an interesting perspective on it. Yeah, how many different ways can the model break?
Yeah, and so what sets a parameter gives nominally normal behavior. When it breaks, what kinds of breaks are there? And when it breaks and you classify the breaks or the flaws into groups, is there some, is a single parameter making the break, or is it a combination of parameters or multiple parameters independently? Again, there's a lot of possibilities. Kind of the traditional way of doing that is you define some metrics. Like in this model here, you might, a Boolean, does it close and lose the boundary or not? And then you run, for every simulation you run and just manually assign by looking at the images or by calculating some metric, what was the final epithelial contact area. A person could sit down and define a bunch of metrics. They'd be very specific to the model. And you actually would have no idea if they're really what you should be interested in or not, but you could do it. The other way of doing it is you go ahead and do, random sample your parameters, generate a whole bunch of runs, generate a whole bunch of either final images or sets of a movie for each one. The movie is a little trickier to deal with. Let's say it's the final image, but then you try to cluster the image, those final images. And the cluster images, I mean, the kind of the modern way of doing it is to run it through a convolutional neural network that's already trained to recognize images.
Yeah.
Right. It's ResNet or something, which knows dogs and cats and cars and planes and all that kind of stuff. But you run your set images through that neural net. You're not trying to train it. You're just trying to get the penultimate set of nodes. And that penultimate set of nodes before the final prediction is just a new description of the image. And in ResNet, I think it's something like 4,000 features, which is way too many. But you can always now start playing, principal component analysis and k-means clustering and try to winnow the thing down so that you get meaningful clusters of images. And hopefully each cluster of images, there's one cluster that's normal result, and another cluster that's they didn't fuse at all, another cluster, the various flaws that can happen. And then kind of work backwards into the parameter set.
Yeah, what you're describing sounds very much like what would be described in genetics as forward and reverse genetics, right? In one case, what we've been doing is saying, all right, we think we know what's important in the system. We're going to knock specific genes out and look for the phenotypes. And what you're describing is more of a shotgun approach. We're just going to do some random mutagenesis.
Yep.
And then see what happens and classify what we get, and then try to go back and look at the code and see, well, if we got this class, where did it come from in the code?
Right.
Right.
Yep.
Yeah.
So I don't know if I can share screen. Can I stop?
Probably you can if I stop.
So this was a quick, early, silly test. So this is some random screenshots out of just randomly sampling parameters and cell sorting. So if you just, just changing the three adhesion values for condensing cell, non-condensing, and the medium, this is a little sample of, I think I did 4,000 simulations, and this is whatever it is, 24 of them. So some of these results look like cell sorting. Some of them, like this bottom at the right, the two on the bottom right, clearly, things are falling apart. Some of them, like this one in the almost upper left, is checkerboard, right? The exact opposite of cell sorting. And then the one to the left of it is actually the beginning of a cell sort. The one problem with cell sorting is it does a whole bunch of stuff real fast, and then it takes forever to actually finish a cell sort. But you see some other patterns here. This one down here that I'm pointing out, looks, I don't know, like a teddy bear or something. So we took this, we ran it through a neural net, took the penultimate layer of 4,000 nodes, decided 4,000 descriptors is way too many, so did principal component analysis and knocked it down to 100 vectors, and then used k-means to sort it into groups. And the kinds of groups we get, I hope it's in this file. So this row is one group. So it's group 11, has 500 members out of the 4,000 simulations. The number on the right, so 13.38, is how far this guy is from the centroid for the cluster. And the first four images are the four images closest to the centroid of that cluster. First four. So these first four are the representative images of the cluster. The last two are the two images in the cluster that are as far as possible away from the centroid. So these are the two worst guys in the cluster. And you see it's giving interesting clusters. And this is a checkerboard cluster. This is on the way to a cell sort. But it doesn't seem to care if it's red on the outside and green on the inside or vice versa. This is another one on the way to cell sort. I use twice as many clusters as it told me I should. That's why I get a lot of duplicate clusters. Checkerboard cluster, et cetera, et cetera. Eventually you get to the teddy bear clusters, and things are falling apart clusters. So I would be really fascinated in your model, because it's so much more complicated than this is, to see how many ways we could break your model. And see how many of those can you look at one of these clusters and say, oh, wait, we know that's a recognized flaw in Palate Fusion. It's not the one we're studying right now, but it's a recognizable flaw. And if you'd be interested in giving it a try, put it that way.
Well, so it would take substantial work to figure out what set of parameters we'd want to even think about including in the mutagenesis, so to speak, right? Because I mean, you could pick all the contact energies, but you could also pick all those biochemical parameters and the coupling probabilities, and there's a lot of parameters.
Yeah, I think I'd lean to just doing them all.
Okay.
I mean, obviously you can't scan them, no systematic. Well, you could maybe systematically scan each one with 2 values or something like that. I would probably lean towards, well, if there's going to be 100 parameters, we'll just sample each parameter from some distribution, and because you're not going to be able to do an exhaustive, anything like a parameter scan is just going to be much too slow. How long does one of these simulations take?
On my desktop, it only takes like 10 minutes or so.
Oh, wow. Okay.
Not bad. Yeah.
So it would be possible to do thousands of simulations.
Yeah.
Okay, well, maybe I'll contact you, and I'm looking for a project to apply this to. I already have one, and cell sorting is nice, but it's super boring.
All right. Yeah, we should talk and see if it would be possible.
Okay, thanks.
Joel, I think you had your hand up first. Oh, can't hear you.
What about now? Can you hear me?
Yes.
Oh, okay. It's always good to hear your talk about this project. Your model is excellent. Every time I look at it, I'm trying to replicate stuff that you created there. And one of the questions that I have is, how was your initial idea about this, what you represent? Because that is the hardest thing, right? As you were talking now, I think is a good segue with what Jin was presenting, which is, do you look first at all the possible cleft palate outcomes, and then you trace back and think about every single agent, their participation in this role? Or do you start with the cell's capacity from omics, for example, and then you roll in the other direction?
Oh, it's a good question. No, I mean, I think we took, I read a lot of literature on Palate Fusion to begin with, and it seemed like there were largely two classes that had been recognized. Either the palate shelves just didn't grow sufficiently, or they grew and met and never fused. And so we sort of set out, well, okay, that looks like two failure modes that we can try to reproduce, and limited ourselves to that as really our target. And then, you know, we included what we had to reproduce that target. I mean, the model seems complicated, but that's what it had to have to really reproduce those two particular targets.
Yep, excellent. Thank you.
There was another hand raised at one
Did
point.
you have a question? You're mute.
I can't hear you.
My question ended up getting answered, but I just wanted to tell Jim, it's funny that you mentioned applying a neural network to this problem, because while trying to think of my hackathon idea, I thought it would be great if there was a feature where instead of having to play with the parameters in an XML file, I could just run a gradient descent algorithm that would continuously guess the parameters for me, and then give me the right ones that achieve the desired result for, at least for the baseline model. So if I'm trying to achieve angiogenesis, for example, then I'd set some kind of condition for success, like each cell is touching at most 2 cells or something like that. And if there's a good result such as that, then the algorithm would say, let's tune the parameters and continuously exploit that.
Yeah, that would be nice. Yeah, we can go through the exercise in a moment. If Joel, you had your hand up again?
Yeah, I did. I had a question, but I was start thinking of the problem that Pete mentioned, and you slapped my mind.
All right. Well, why don't we take a look at some of the exercises, and then we can we can stop for questions as people have them. Let's see. The best way to do this is probably to just share my desktop again.
While you're putting it up, I remember.
Okay.
About the chemical interaction there. Do you have the literature saying that is the exact mode of action, or it was something that it was hypothesized, this interaction between the EGF receptor, and instead of like capturing the TGF beta, because those would probably produce the same result, right? There's multiple ways.
Right. That was based on the literature, right? So it's just what people have been able to track through. It was really well established that it acts through the aryl hydrocarbon receptor, the AHR part. But then the downstream effects of that can be pretty widespread. There's lots of different things that can happen downstream of AHR. But there were the best indications in the literature were that it acted through changing the expression levels of the EGF receptor. And so that's where we put it into the model.
Great. Thank you.
All right. So let's see. I will start with this Nucleate Compartments 1.cc3D project here. And I'll give you a little bit of a tour of what's in this, right? So this uses a combination of XML and Python. There is an XML script for it. I'll open that up here. That really defines all the basic parameters for like contact energies and things like that, and whether we're going to use volume and surface plugins, right? All of those are defined here, and the contact energies are defined, and the blob initializer is defined to really just set things up. So there is an XML file that's present. There is a Nucleate Compartments.py file that's really very simple, and its only job here really is to call the Steppable file where the real functions are defined. And so you can say it imports this Nucleate Compartment Steppable and registers that and make sure that it runs every time. And the real heart of everything is in the nucleate compartment steppable file. Right? So if we take a look at what's in this one, the start part, right? So call before Monte Carlo step 0, it just goes through every cell in the cell list and sets the target volume and the lambda volume for each one of those, right? To be what it was initially. And then the step one here, okay, what it does, let me run this first and you can see it, and then I can explain it a little bit better, what we're trying to do with it. So let me open this in the player. Take a second to do that. Oh, all my cell type colors changed. Now let me close that up, restart it. All right, so you see a big blob of blue cells there. Those blue cells were of type mesenchyme, and then some green cells popping up on the outside of it. And the idea here is to let the system differentiate over time based on the conditions that each of the cells have. Okay? And you see that over time, it's going to fill up that outer layer. It takes about 1,000 Monte Carlo steps for all of the outer cells to become green. We got 1 or 2 holdouts there, and they'll eventually probabilistically turn green. There we go. So now we have a complete epithelial layer around the outside. Now, to show you what's actually behind that and what it's doing to do that differentiation as a function of time as it goes through, right? So it loops over the cell list for cell and self.cell list. We have a function that's going to figure out whether the cells are in contact with the medium. So we set this Boolean at first to say, all right, it's not in contact with the medium, and so that's going to be false there. Okay. Let me make this a little bit bigger so we can see all the lines there. We set a probability to differentiate because we're going to have this change of cell type be probabilistic. And this line here, where the probability is changed based on the frequency with which it's called, this is based, this mathematical structure makes sure that whether we call the steppable every 10th or 20th or 50th, that the rate at which cells differentiate into epithelium on the outside stays the same. So it lets you troubleshoot things by calling it very seldom, right? And then you can do production runs where you ramp up the rate at which you call it, and it shouldn't change the behaviors, right? And so here's the heart of it, where it's going to figure out whether to actually differentiate a cell or not. And James and everyone, correct me if something has changed in the way that neighbor lists are considered, things like that.
No, I don't think any of our basics have changed much.
Okay.
So everything should be working the way it used to.
All right. So in this case, we're going to use this list, self.getCell neighbor data list. And what it's going to return is all the neighbors of the cell we're currently considering and the area of the contact between those two cells, right? It returns this tuple, and so we'll assign both of those into there. And so this if neighbor cell would let us do something based on the identity of the cell, but we're just going to pass because we're really just asking, is the cell in contact with the medium? And medium is defined as essentially a null cell. So if we say if neighbor cell, that's going to be false if the neighboring cell really was medium. And so we'll go down here to the else part of the statement. And so if the neighbor cell was medium, and so this was false, we're going to say in contact with medium flag equals true. We're going to use this Boolean and just flip it and keep track of whether the cell's in contact with medium based on that flag. And then if that flag is true and the cell's current type is mesenchymal, then we're going to randomly differentiate it if some random number we pick is less than this probability to differentiate. And there we differentiate it by just changing the cell type to epi. Okay? And it'll just do that each time as it goes around. And what you notice as it does this, I'll play it from the start again. Colors changed, interestingly. So this time the white cells were the mesenchyme, and it's changing into these cyan cells on the boundary. And those fill in. And one of the really nice things about doing it this way is that you get differentiation in place, which is more like what happens during development, but you also get maintenance of the epithelium, right? Suppose some cells died or a gap opened up, well, new epithelial cells would differentiate and form on the boundary there, right? And so it allows you to have sort of active regulation of having a polarized epithelium. Well, not polarized yet, but having an epithelium that's in there. All right. Does anybody have any questions about that model before I sort of let you loose on the exercise, right? The modification of it to do the next thing? All right. So the next exercise then is written in here. So build on this solution so that after a cell is differentiated into an epicell, it polarizes to have apical and basal compartments with different properties. Yeah, there we go. The basal compartment can be the original epicell type, so we can stick with that. But we need a new apical domain. I often use epi and then a lowercase a, for example, to represent it being the apical compartment. The goal is to generate the polarization, maintain it, and keep the polarized epithelium well organized with all the apical domains on the outside in contact with medium and all the basal domains on the inside. All right, so that's the goal. So we want to differentiate not just epithelial cells, but how you would have apical and basal parts of those epithelial cells. So sort of the pseudocode to give you an idea of what you're thinking, how to think about it, and then we'll get the actual code into it. We want to loop over all the cells. And then if a cell is of type epi, we want to check whether the cluster to which it belongs has an epi-A compartment. If it doesn't, then nucleate one on part of the epi cell's surface that's in contact with the medium. And that nucleation will require creating a new cell of type epi-A, assigning some lattice sites to it and setting its volume and surface energy targets. Right? Some useful things, functions to help you get pixels in contact with medium. That'll help you both determine whether a cell's on the outside and whether where you can initiate or nucleate an apical compartment. And they'll help you. So I'll let you give that a shot. You can ask questions as you go and work on it. If you get that done really easily, challenge is to add a line that randomly kills off a few basal epithelial compartments and see whether you can add code that'll restore those, right? So it's actively maintaining the polarized epithelium. Yeah, James asked a question, right? Are those of you, can you open up this code and get it to run properly on your ends? At least the nucleate compartment 01 to start with. All right, good. So I'll shut up and not distract people too much, but I will say one thing, that you'll have to make modifications in two different parts, two different files here. right? Because we need an epi-A cell type, you'll have to do that in the XML and define the contact energies for it there, and then you can define the functions for how you make one of those during the simulation in the Steppables file. All right, while folks continue to work on it a little bit, I'll show you what the result should look like, what we're talking about making sure it's polarized. We'll just let this run. So you can see there the green and red popping up on the outside. Those are the apical and basal compartments of the epithelia cells. And it's filling in. And we have a few that are missing apical compartments. I have to check. Those are not filling in. Yeah, Nucleate O2 is the solution for those who want to take a shortcut. We'll go over that in just a second. We'll give people like another minute, and then we'll talk about that one to wrap things up. All right, let's take a look at the nucleate compartment's O2 code and how we did this. And I will talk about how you identify the cells with missing compartments. It would be really nice if there was a shortcut to do that, right? Just a little plug-in. That would be something that would be very helpful to have. But we can take a look at this. So if we look at the XML, right? We had to add another cell type. So I added an epi-A cell type in there, type 3. And we had to add it down here to the energies. And so you'll see several lines that have epi-A. Contact with medium is very favorable. Contact with mesenchyme is not favorable. And contact with itself is sort of in between. So it wants to end up on the outside of the patch, wants personifying it here. Energetically, it's lower energy to be on the outside of the patch. But notice that the external energies are different from the internal contact energies, right? And so internally, it's really favorable for an epi cell to be in contact with its own epi2 compartment or epi-A compartment. Notice that that's much more favorable, right? Internally, epi-A is an energy cost of 2. Externally, epi-A is a cost of 10, right? And so this makes it so the most favorable alignment is to have an apical compartment attached to its own basal compartment, and then those side by side with apical and basal of the next cell, right? And those are the only differences that took place here in the XML. We made a new cell type. We gave it the appropriate contact energies. And then in the Steppable file, we had this function before, get pixels in contact with medium. So I won't go through that in detail, but it's a nice thing to be able to get the pixels that belong to a particular part of the boundary. And what do we do down here now? All right, so we're looping over every cell in the cell list. We define the flag and the probabilities differentiate just as we did before. This first part, this little for and this if statement here, determines whether we should differentiate cells from mesenchymal to epi. It's exactly what we had before. The second half, this is the code block now that will do the compartmentalization and check for matching compartments. All right, so if a cell type's epi, we'll start there. We're not going to bother with this if the cell's a mesenchymal cell. We're going to first set this polarization flag false, and we'll change it to true if we figure out the cell has been polarized already. We get the compartment list. So this is get cluster cells of the cell. So it's basically the list of other compartments or cluster, or the list of other cells that are part of the same cluster. And there's some different ways to describe this. A cluster is really the same thing as a compartmentalized cell. So we're going to loop over the compartment list. We have to do this a little oddly because compartment list isn't really a Python list. So you can't just do for compartment in compartment list. You have to do it with this index. But we're going to modify the polarization flag, right? And this polarization flag equals polarization flag or compartment list I.type equals epi. Make sure the polarization flag stays false unless there is an epi compartment in the cluster. As soon as there is an epi compartment in the cluster, the flag turns true and it stays true thereafter. And so this is then checking, all right, if that flag is true, well, great. That epi cell already has a complementary epi cell, and we'll print that out just to keep track of it. If it's not polarized, if we get here and polarization flag was still false, well, we'll print something out about that. We will make a new epi cell, right? So we'll self.new cell there. We will get the pixels that are in contact with the medium, and that's what we use that function up there above for. And from that list of pixels that are in contact with the medium, we're going to assign all but 2 to the new epi cell, right? You need to assign more than one to make sure that the nucleation takes. So we assign all but 2 to the new apical compartment, and that's all that this is doing. And eventually, right here, this self.cell field XYZ equals new epi cell. We're assigning those lattice sites to that new cell that we just defined. Then we want to make sure we define some target volume and lambda volume and target surface and lambda surface for the new cell that we just created. And then a couple of reassign, a couple of print statements to track what we're doing. And a really important one down here, this reassign ID flag. So we have to take the new cell that we just created and make sure it gets assigned to the same cluster as the cell we were checking initially to see whether it was compartmentalized. So there's 3 steps. We have to make the new cell, assign some lattice sites to it, and then make sure we set its cluster ID to match what the original cell's cluster ID was. And that way there are 2 compartments in the same cluster then. Okay. Questions? We can do the right one there. Let that crank up and run again. And you can see when one of the epi cells pops up, it most of the time immediately makes an apical compartment to go with it, right? The little red cap that goes on it. Although there's 1 or 2 cells that I'm not exactly sure what's going on with them that don't have an apical compartment and don't seem to be making one. They should. Yeah, so I find the compartmented cells to be really critically important. We were just developing a wound healing simulation this summer, and we actually had to have both apical, basal, and nuclear compartments. It turned out to make that one work correctly. Gives you a lot more parameters you have to keep track of. And it would be nice to have a way to designate that in an easier table, but once you get those set, then the compartmented cells is a really powerful extension of what CC3D can do. Yeah, Joel.
Can you give an example of like the reasoning behind the compartmental cells in that case? I mean, I'm imagining that you only have one cell type on your epithelium, or you're just considering one cell type on your epithelium. Is that correct?
So we are considering one cell type on our epithelium, but we want, in essence, we want a sheet of cells, but we want to do it in 3D. And so to keep a group of cells behaving as a sheet, one of the most straightforward ways to do it is have compartmental cells that have apical and basal compartments with different contact energies. And it will organize itself into a sheet and be very stable as a sheet over long periods of time.
Thank you.
Yeah, because if you didn't do that, it's going to round up, right? It's going to want to cluster as just one big pile. And so to keep it organized as a sheet, that's largely one of the reasons that epithelial cells in biology do have to polarize, is that they have this sheet-like structure, and maintaining that is and polarization is a key part of maintaining that. Yeah, getting the parameters right so you have 3D epithelial tension is tough. It is doable, but it's a little brittle, I would say.
I was going to say the other thing that's an issue is, I mean, we often will do epithelia where you'll have apical, basal, lateral compartment. And then the apical compartment, you'll actually have two subcompartments which you use for junctional adhesion with the neighbors.
Mm-hmm.
And that way you could actually have apical constriction easily, because you put a link within the cell, you put a link between cell, center of apical compartment, lateral side of apical compartment. And that works very well. But if you want to do it in 3D, then those little links that are going between the two neighbors is you have a lot more neighbors, and so you wind up having to have a lot of compartments in your cell.
Yep.
If you just have apical, basal, and lateral, then going from 2D to 3D doesn't add the number, doesn't change the number of compartments. But if you actually want to put the links in between the specific neighbors, then it becomes a pain in 3D.
Yeah. Yeah. No, agreed. Yeah, so I hope everybody found that useful. If questions come up along the way as you're trying to do these things, you're more than welcome to email me, and I will chime in where I can and help as much as I can, and refer you to James and Jim and all those guys where you run into the edge of my knowledge. So,
and I encourage everybody to try downloading and running the full simulation. Shane made the effort a year or two ago to update his simulation so it would run in current CompuCell. And it's really impressive. It runs very fast, and it's a beautiful simulation. And he's been very generous to allow us to share it with you.